I'm going to now introduce Frank Gu from the University of Waterloo. Frank, are you on the line?
Yes, I am. Hi.
Great. And I see your presentation. I guess so I'll just turn over directly to you, Frank.
Thanks, Mike. Hi, everybody. My name is Frank Gu, and I'm coming from University of Waterloo. So if you can go to the next slide. Thank you. So the project that I'm working on and leading here is titled Development of Root-Targeted Delivery Systems for Controlled Release of Water and Agrochemicals for Ornamental Plants. So I quickly introduce myself, Frank Gu, and in my team we have 4 people. Drew Davison is a graduate student, Mohit Verma was a grad student, and then we had 2 co-op students who were helping us along in the past year. So the overall objective of this proposal is to develop a delivery system that can provide on-demand release of water and nutrients to sustain ornamental plant health. So the idea is pretty simple, is that if we can optimize a design that we had in our previous study, then this device could be used for a variety of applications for the ornamental and turf growers, and especially in the sense that we can hope to reduce the total amount of nutrients, so fertilizers, needed without compromising the plant health. At the same time, and the gel will serve as a depot to release water on demand as well. So based on our previous study, we have shown we can able to drastically reduce fertilizer and water requirement for greenhouse plant from lettuce, tomatoes to green peppers. And we wanted to apply this technology for ornamental culture growers and related applications. And the specific objectives here is really to customize this for this use. And what it really means here is to optimize the methodology that we can use to implement the delivery device. And we call it RTDV. RTDV stands for Root Targeted Delivery Vehicles. And we want to see that how to optimize the delivery vehicle that are relevant to the soil mix that are for the growers here. And this is talking about with respect to the size, the loading capacity. So the loading capacity relates to the amount of water and fertilizer required for each individual applications. And second is to looking at a total reduction on the nutrient required without compromising the plant growth efficacy. The third objective here is to look at and carefully examining how does a delivery vehicle degrade with respect to the soil conditions, and to further tailor the delivery device so that when the application is finished, the delivery vehicle itself sort of fell apart and naturally absorbed by the soil, as opposed to it becomes a permanent structure somehow in the pot. So that's sort of the area of focus. And finally, another strategy is to formalize the IP and the commercialization pathway. So we have done that for plants, for food applications, but this is the first time we begin to look at it for ornamental and horticulture applications. In terms of the expected deliverables, our focus on number one is, can we reduce the amount of water required, and by how much? So the first delivery expected is that the RTDV will provide on-demand delivery, and essentially we're hoping to cut fertilizer and water by about 50% without compromising the health and growth. And the number 50% came from our previous work on tomato and green pepper, in which case we showed far greater than 50%. So we set 50% as a benchmark, and this is to do with, in consultation with the nursery that we've been working with, that if we can reduce it half, that's a good number to start. The second part is to demonstrate that we can maximize the efficiency of usage. So when we say root targeted delivery, we wanted to demonstrate that the root, in fact, is being entrenched by the delivery device, and so it provides that on-demand delivery. That is completely different than what's currently available in the commercially available fertilizers. So the main difference we want to show is that what we want to show here is that the currently available commercial release fertilizers requires typically undergoes a burst release of fertilizer. So the fertilizer gets released very quickly, has a dump effect, or it has a change in pH or local environment because of the delivery vehicle itself, which somehow it could interfere with the plant growth. So what we want to show is we can do all the things of delivery that we promised, but at the same time don't have the undesirable side effects that we're seeing in the controlled release fertilizers. And the next deliverable that we hope to achieve is that if we can deliver nutrients and water, it's not a far stretch to consider expanding the application into not just nutrients, but also therapeutics, antifungal, or other herbicides, insecticides that can be co-delivered at either a similar rate or a different rate that can further reduce the environmental impact on the overdose on the soil itself. The next part is, we could do all this, but what is the actual cost? So this is actually what we are trying to finalize here by the end of year one. And then with the cost reduction comes with increasing the throughput, so we can do this not just providing 100 plants or 1,000 plants, but we can provide it on a larger scale. So those are the questions that we hope to deliver within the next 2.5 years. So the way that we divided the objectives is the following. For first, year one. Year one is focusing entirely on nailing down the manufacturing process itself. And I'll talk about this in the challenges coming up in the next few slides. But essentially, we proposed a 5-year program, and now it's being squished into 3 years. So what we thought is that in our 5-year program, we have the growth efficacy demonstration throughout each year. So we provide many different plans. In order to still fulfill all the milestones, deliver all the milestones, but at a compressed timeline, what we decided to do is revise the objective. So year one, instead of focusing on delivery efficacies, but we're focusing purely on the manufacturing process. So this way will give us the ability to grow a much more different plants, so that we're not getting a setback in the following years if we're going for a greenhouse trial, but we're still trying to figure out the scale-up process. So the year one focuses on making the delivery vehicle, characterizing it, and ensuring that we can provide a sufficient quantity for the future studies. And then finally, because we're making the delivery vehicles ahead of time, this will give us the ability to measure the cost and the cost analysis and the throughput, but also give us the ability to look at the degradation profile of this, or the storage shelf life, because now we have to provide enough material that can last us for the following summer or the summer after. So this is where what we're looking at strategies to prolong shelf life. In addition to the compressed timeline, one of the biggest challenges we had this year was that we had originally a PhD student who was all ready to go on this, but because the timeline wasn't right, so in fact he ended up graduating just before the official launch of this project. And so to compound the additional problem, the other student who were trained by the student who graduated had a very unfortunate, unexpected medical leave from September last year, 2014, until this year. So that actually made the problem a bit worse, because we supposed to have 2 people and one training in the other, but because of this timeline compounded with the medical issue, we end up having to scramble. And the way that we resolve this issue is, instead of hiring additional research technicians or additional graduate students, which would take a much longer, a steeper curve for doing the production, we decided to have the student who graduated remotely trained 2 of temporary staff from a co-op staff that worked to make up the time that we lost. And fortunately, we got everything back on track, so now everything's proceed as we planned. So the challenge has been resolved. And in terms of the objective, the year one, in terms of making the RTDV and the characterization, it essentially focuses on synthesis, and then we do testings and trials, and then provide a refinement to provide additional improvement on making this gel. So I just wanted to maybe spend 5 minutes to talk about the update we have to date. So first is that we have produced the vehicle. We have performed all the physical chemical characterizations. We have also begun an accelerated degradation study in soil to look at the ability to perform. We have also begun the scaling process, and I'm glad to review that we used to be able to make this on a batch basis, so it really becomes very labor intensive, and it could suffer, the device itself could be at a risk of having a batch-to-batch variation. But we have successfully migrated the batch synthesis to a semi-continuous, and now we're on to almost a continuous process, so that we can begin to make the same delivery vehicles, but at a much higher throughput. So this will give us sufficient quantity to last for a much bigger trial for a longer period of time. So now what we're doing is to finalize all this for beginning to all the growth studies for year two. In terms of results, I wanted to show you there was, in terms of the RTDV synthesis and characterization, the question I guess a lot of you might have a question is, if we have shown a fairly good growth and efficacy in tomato and pepper, why are we spending more time to remake the synthesis of hydrogel? To answer that, one of the answers is that we wanted to tailor the soil media that would be specific to ornamental plant growers. That's one answer. The second answer is that we found there was a faster and a better way to do it. So the original study we have done is using ionically cross-linked hydrogels. So these are the gels that are fairly affordable and cheap, and they're made to become a gel by adding additional ions. And the advantage of doing this is that the process is simple. Everything can be done in ambient condition, and the safety and efficacy have already been proven. But the disadvantage is that it's the material, in fact, couldn't last very long in soil. So for turf applications, this particular device can last for about 31 weeks in soil, and so the water can be reloaded once a week without compromising the growth efficacy. So it lasts for about 3 months. It lasts about 3/4 of a year. But that's certainly not the same type of duration that we can look for ornamental or for bigger applications. So last year what we did is to switch the ionic cross-linking chemistry to chemical cross-linking chemistry. And as a result, we have developed gels that has a much better ability to resist degradation. And it has the same type of loading capacity and performance, but now it can last, instead of lasting for 31 weeks, we now start to see that it can last for much longer. So our degradation study is still ongoing, but it can last at least 1 year at this moment. So actually, the next slide is the work we did on the ionic hydrogels. I won't spend any time on it because this is the device that we were trying to improve on, and so I'm going to skip that slide for the time. But the performance is very good, and we show this growth on wheat, also on peppers, that we can provide the same kind of growth and at an even faster rate, but using a much lower loading of fertilizers. So next slide, please. So then we switch to the covalent cross-linked hydrogels, and this is a glutaldehyde. This is a very commonly used cross-linker from anywhere from medical applications to industrial applications. So the technology for cross-linking itself is being well proven. And the gel material and handling and properties becomes much easier to work with and more compatible for long-term usage. the overall challenge we had to make for the chemical cross-linking hydrogel is that the cross-linker themselves could be toxic to the seed growth and overall plant health. So the challenge is that we got to make the gels, and we have to remove all the unreacted or unused cross-linkers so that there's zero residues behind. So this is where we spent most of our time last year, is to tweak this chemistry so that it can perform, but be able to completely remove the cross-linking material, so the gel is becoming a gel and removing any potential toxicities inside. So anyway, we had several ways to do it, and I don't want to bore you with all the details, but we found out a way that we can make the gel, remove the cross-linkers. So this has gave us the way of making a clean gel. So now this delivery vehicle is a delivery vehicle without fertilizers. Then what we do is we developed a reverse loading strategy that we can load the fertilizers in. So this is another something new that we developed here compared to the ionic gels, where the fertilizers were loaded into the gel during the gelation process. Here we're making the gel and then we load the fertilizers. So this gives us two advantages. First, we can completely remove any type of cross-linkers without compromising the site reactions with the fertilizers. Because what we showed is that if you have the cross-linker and the fertilizers, they will react with each other, which is not surprising. I mean, there's a lot of amines in there. And that would be not desirable. So we found a way to make the gel to avoid this issue by making the gel and remove the unreacted cross-linkers, so there's no cross-linking reaction or cross-contamination between the cross-linker and the fertilizers. And the second advantage we found is that because every plant that we want to examine have different needs for the fertilizers. So some of them requires a high concentration of one element but not the other, whereas other plants may have different needs. So if we have a delivery vehicle that have just one type of fertilizer composition present, that's not very attractive to the growers. So by doing this new approach, now we can begin to unload and load the nutrients that can be customized and tailored specifically to the need of individual growers. So by doing this, it gives us these two advantages. So we haven't made all the biological characterizations because of time. We've been trying to catch up because of the student issue. So what we did is to produce a quick growth trial to show that the material is not toxic and that the performance is well demonstrated, so that we can continue on with the scale-up production. So we used instead of using a plant that we don't know, we decided to choose a plant that we worked before. This is a canola, and we begin to demonstrate the gel growth loading with no amount of fertilizers compared to a commercially available controlled release fertilizers, and the results were quite promising. So the question that you might have is, how come the commercially available fertilizers perform so poorly? The reason is this type of soil media we chose here. So we specifically chose a soil mix that is highly porous. The porosity provided a mean to quickly eliminate or remove all the fertilizers that released by burst effect. So when the commercially available controlled release fertilizers got burst, so the fertilizer gets released, they will release a mass loading quantity, but then there's no time release rather than instead of a triggering release. So in a highly porous media, all the burst release fertilizers will be quickly removed right away. So this will compromise the plant health. So in our case, we designed the experiment specifically to demonstrate that in a highly porous media, so in other words, if there's no nutrient retained in the media, the plant has to be able to survive on its own by having a slow release media present. So this is our study to indicate that this newly formed hydrogel is able to release the fertilizers at a very slow rate. Had it released a very fast rate, the plant wouldn't have grown because it will get the excess fertilizer will get washed away. So this has to be going very slowly. And second is it has to grow at a point that's very close to the plant roots itself to provide that localized effect. So this particular study is not designed to compare the growth rate, but simply to demonstrate that the hydrogel itself that we make are able to load fertilizers, release them at a slow rate, and the growth efficacy can be shown in this demonstration. Next slide. We also begin to look at degradation studies. So this is the first set of degradation study we have done to look at the number of days it's been done. One thing I should have add a data here, a description here, is that this is the accelerated degradation. So this is not a natural degradation, but accelerated. So the way that we perform accelerated degradation is by increasing the acidity of increasing the acidity, decrease the pH of the soil media to further accelerate the carbohydrate degradation from the delivery vehicle. So we show that, in fact, what this is trying to show is not about how quickly it degrades. That's something that we're going to report at the end of the year. What this graph is trying to show is that with the degradation environment, all the component of this delivery vehicle can be degraded and removed completely. So again, we're not looking at specifically paying attention to the curve of degradation, but simply to say that after we do all the cross-linking, the material itself is still degradable. So that's all we wanted to show at this moment. And the science behind the rate of degradation and the kinetics, that's something we're going to show later on. So the works are ongoing right now in addition to the accelerated degradation study to understand the potential shelf life. Other things that we are working on includes doing the cost evaluation on the scale-up process. So earlier I mentioned we have moved to a continuous process. So we want to look at, from a modeling, how quickly can we build into a pilot plant, and when this project is finished, to meet the demand possible for the growers' demand to the production through rate with the cost in mind. The other thing that we are looking at is, we're going to make these gels. Ideally, they should be able to sit at a shelf condition for at least 2 years. So how does moisture or potential temperature or other factors that could impact the degradation profile? That's something that we are looking at it right now. In terms of year 2, once we have completely the degradation studies and characterization and scale up, next year we're going to focusing very heavily on the growth studies, and we welcome any advice for additional plans or substituting plans from what we proposed here. And then we're going to throw in, from a chemistry side, we're going to add additional therapeutics to look at a combination of delivery of nutrients and therapeutics at the same time. Year 3, we're going to focus on more complex plans, as well as looking at additional delivery systems. And we originally put the effect of soil conditions in RTDV degradation in year 3, but based on what we have seen so far on our data, it's very likely we'll move the soil degradation study to year 2, just to give us sufficient time to provide all the characterization on the degradation itself. In terms of knowledge transfer, the way that we're thinking about communication is this technology. We're hoping that as many people use this technology as possible. But we wanted to do is to develop a technology that we can tailor to our growers. So rather than me saying this is how much nutrients we need, we want to listen to the grower, to the specific plants and the specific soil they wanted to grow, and then we can design a growth trial specifically to meet that demand. So this is particularly how we switched the strategy from growing to do all the growth trials in a 5-year study to a 3-year. So we focus in year 1 to develop the vehicle, build up the platform, and then we want to work with as many growers as possible to customize this delivery vehicles to the specific needs of their specific needs. In terms of communication-wise, we'll focus on publication. And then also, once we finish the continuing process for production, we want to develop a 2-page non-confidential disclosure pamphlet that we can circulate to all the growers to say, here's what we can do, and let's see where we can take it from here. So that's all the slides I have. And Mike, sorry I took 4 more minutes than my allotted time, but thank you for your attention.
That's good, Frank, and thanks very much. We have a couple of minutes for some questions, if anybody has some burning at the moment.
Yes, I have 2 questions. This is Guillaume Grégoire. First, what do you expect the product to look like physically when it's going to be on the market? Is it going to be a gel like that, or I'm trying to figure out, seeing a grower, how he's going to use it, or somebody in turf. You mentioned you want to do a trial with turf grass, so how do you spread that product basically on the plants?
Okay, so the way we did it, we had two methods. So first is make them into a bead shape, and the bead shape is made by batch reactions.
New participant has joined the conference.
The new one we're trying to do is to make it into a pellet shape, so like a small little pill size pellet. This way, it would allow us to make a whole slab, and then we could chop them into small pellets. And that's how we can make it into a continuous production.
And these pellets, they're made of gel, like they're not dry. They're
Oh no, the picture we show you is the fully swollen gel.
Okay.
The actual gel itself is about 1/10 of the size.
Okay, so it's going to look basically like current fertilizer pellets that are available in the market. Physically.
Slightly bigger. So imagine a Tylenol pill. I mean, that's sort of the shape we have. So it's about 5 millimeter in diameter of pellets.
Okay. Second question, you mentioned wanting to trials with fungicide. If I understand the technology properly, basically the fertilizer stays in the gel until it's required by the plant. So are you afraid that the fungicide will actually stay within the gel and not be able to affect potential fungus that are in the soil? Or do you have any preliminary results that show that it works basically?
We show load fungicides, excuse me, into the gel, excuse me, and we have not considered the rate of release because we simply don't have a benchmark to compare to. One of the ways that we have strategies to look at is to encapsulate the fungicides into a gel and then load it into the delivery vehicle. So that way it doesn't interfere with the normal growth studies. But the details of that's something that I would love to learn more from you or other growers on exactly what's the best way to tailor your demand.
Okay, thanks very much, everyone.